My name is Robert Morhard. I'm a 6th-year grad student in the Ramanujan lab. During my first year, Nimi and I had a series of conversations about how there were a lot of increases in diagnostic and screening capabilities, specifically for cervical cancer was our focus. And there were kind of like a dearth of options for treatment in resource-limited settings. So what we wanted to work on was making a treatment that would be compatible for those settings. And so we looked at a lot of different options, and ethanol ablation stuck out to us because it's very cheap. It doesn't require a lot of hard-to-supply consumables like other techniques. Tumor ablation refers to techniques that destroy tumors but don't remove the cells from the body, so they destroy them like within the body. So some techniques use heat to kill cells, some use really cold temperatures. Our technique uses ethanol, so you inject ethanol into a tumor and it kind of dissolves the cell membrane and denatures a lot of the proteins. But as we looked more into it more, we realized part of the reason it had been phased out in the past and replaced with more expensive technologies was because it was hard to kind of control the treatment region. So if you inject it into tissue, you might kill a lot of your malignant tissue, but you could also kill some of the surrounding tissue because you don't know exactly where the injection is going. And so we've been working on kind of improving that injection process.
There's a lot of things to control in this. Because in the cervix, you basically want to be able to preserve the function of the cervix, which is important during pregnancy and delivery. And so you only want to kill those top layer of cells that are harmful and leave the rest of the cervical muscle intact.
Conventional ethanol ablation, you inject pure ethanol into tissue and it just kind of goes all over the place. With the method that we're working on, when you inject it, it forms this gel, and ideally that gel will kind of trap the ethanol right around the injection site.
When you add water, which is present in tissue, to our ethyl cellulose ethanol combination, you get this gel formation. And so that essentially helps keep the ethanol where you've injected it and allows it to slowly leak out and release over time. And as cells are exposed to high concentrations of ethanol, they will actually die. Whereas if you just had a flash in the pan with just injecting pure ethanol, you wouldn't necessarily get the region of cell necrosis that you're aiming for.
We're basically proposing a modification of a technique that was used in the 80s. And so after like 30 years, there's been a lot of changes in tumor ablation and just cancer treatment as a whole. And so people don't always want to go back to that. But I think one of the downsides to that is that those techniques that are 30 years down the road are really not compatible with resource-limited settings. And so they're solving and working on a different problem than we are. So they bought it from like a pharmaceutical. I think our group tries to be really open-minded about these sorts of problems. And so even though something on face value might seem like a bizarre idea, I think we try to think of why specifically is this a bad idea, and if you can't really name any reasons, then maybe you should look into it further. And I sort of feel like I've been in that stage with this project for the past 5 years, where it's like, this doesn't seem like it would work. It's not, conventional, but then the more into it, it's like, oh, why wouldn't it work? And I still haven't really found a reason why it wouldn't work. As a researcher, the freedom to work on whatever you think are the biggest problems and the techniques that you find most effective is really crucial. So I try to think of my goal not of getting this treatment into clinics, but just properly assessing whether or not it is a good idea. And so I just try to stay as objective as possible and just accept whatever the reality is as it is and just move forward from that. It's hard to not cheer for the thing that you're working on, but you never know that the failure that you had might be instrumental to someone else's or even your future success. So you just have to report everything as unbiased as possible and just hope that eventually it'll lead somewhere worth going.